This is Izzy Walker, and I'm going to be presenting my bacterial unknown project. So I received unknown organism number one, which turned out to be Klebsiella pneumoniae. So I'm going to be walking you guys through the whole process of how we identify an unknown, and specifically how we ID Klebsiella pneumoniae. Take a look at our sample and our requisition. So we've received a urine sample, and the requisition says that we've got a diagnosis of a fever, and they're going to want a aerobic culture. Now there's a lot of media that we can get into, but I specifically want to focus on our blood agar and our MAC, and so we're going to do a 4-quad streak because we want to isolate our colonies, and then we're going to incubate that for 18 to 24 hours at 35 to 37 degrees Celsius. While we're growing our culture, we're going to want to do a gram stain right alongside that, and we're going to actually want to look at that first. So let's say we stained it and we're looking at it under the microscope and we see a gram-negative rod. Now immediately that makes me think that we're working with an Enterobacteriaceae, or an enteric for short, and we know that all of our enterics are going to be oxidase negative. Now let's look at our blood agar plate, and I'm seeing very mucoid, wet colonies. And this is going to be our polysaccharide capsule, which is just a virulence factor, and it's going to block engulfment by immune cells like macrophages and neutrophils. And then let's look at our MAC plate. Now we see pink colonies, which is immediately going to tell us that we have a lactose fermenter. Now, not all enterics ferment lactose, but we're going to want to keep that in mind as we move forward. We're going to start our biochemical testing for our suspected enteric, and we've got our IMVIC and our set of 5. And our IMVIC is going to be our indole, our methyl red, Voges Proskauer, and our citrate. And this is looking for chloroforms, which are a subgroup within the Enterobacteriaceae family, ideal for narrowing down our lactose fermenters. And then our set of 5 is going to be our TSI, our lysine, urea, MeO, and our citrate. And these are typically done after the IMVIC, and these will help separate organisms with similar IMVIC profiles. Now the MeO tests are interesting. These are like a multitasker test, and they typically run parallel to the IMVIC, and it complements the IMVIC by adding motility and ornithine decarboxylation. Now, just a recap, we're looking for an enteric lactose fermenter, and we're going to want to confirm that with our oxidase test, which should be a negative, and then we're going to want to look for other sugar fermentation. Now, our first test will be our oxidase test, and this is just looking for the cytochrome C oxidase enzyme. And if that is present, it'll turn the area dark purple within 10 to 30 seconds. And in this case, we have a negative, so we are working with an enteric. Into our IMVIC tests, which are indole, methyl red, Voges Proskauer, and our citrate. And so let's start off with our indole, and this is asking if we can break down the amino acid tryptophan. And so we're going to add our COVAX reagent after our growth, and it's going to be positive if there's a red ring at the top, which means that indole was made, and in this case it wasn't, so it's going to be a negative. Next is our methyl red test, and this is asking if our organism can make strong acids from glucose, and so we're going to add our methyl red indicator, and our positive is going to be a red color, which means that there's lots of acid, and in this case it's going to be negative, which is going to be a yellow color, which means that there was little or no acid. Next is our Voges Proskauer, and this is asking if our organism can make neutral products like acetone from glucose. So we're going to add our VP reagents, A and B, and it's going to be positive if there is a red pink color at the top, which means that acetone was made, and in this case, this is going to be our first positive. And lastly, we're going to do our citrate test, and this is asking if our organism can use citrate as our only carbon source, and we're going to grow it on Simmons citrate agar, and it's going to be blue if the citrate was used, and in this case, we have another positive. Now, looking at our IMVIC profile, this is classic for our Klebsiella pneumoniae, and this is a really good test to differentiate that from E. coli, which is going to have the opposite profile. So this isn't the only organism with the same profile. We've got our Enterobacter aerogenes and some strains of our Spiraceae marcescens, which also has the same one, but the Klebsiella pneumoniae is going to be the most likely case. Let's move on to our set of 5, and let's start with our TSI. Now the tubes in the back are going to be our uninoculated, and the tubes in the front are going to be after inoculation. So our TSI, or triple sugar iron, is just going to check for sugar fermentation, and based on the entire color change, it's going to be an acid over acid, and we see a space in the bottom of the tube and bubbles, so that's going to be our gas production. Now there's no black in the tube, so that means that there is no H2S production, which is just our hydrogen sulfide. Next, let's move on to our lysine, and that's going to be alkaline over alkaline, and there's purple in the slant, which means no deamination, and then there's purple in the butt, which is going to be a positive for decarboxylation, and again, there is no H2S production because there's no black in the tube. Then let's move on to our urea, which is just checking for our urease enzyme. And since there's only pink in the slant, that's going to be a weak or delayed urease activity. And then lastly, our myotube, which is our motility, indole, and ornithine. And so after we stab our media, the motile organisms are going to spread away from the stab line, and we don't see that in this case, so it's going to be non-motile. And then our indole production, just like last, there's going to be a red ring at the top, and there's not, so this is going to be another negative. And then lastly is our ornithine decarboxylase, and a positive is going to be purple throughout the entire media, and we see yellow at the bottom, which is going to also be a negative. So now we're going to move on to our automated tests, and not only is this to identify our bacteria, but it's also to determine which antibiotics are going to work best. And it's going to include a panel, which is just a tray with wells containing dried biochemical antibiotics. And it is going to include a purity plate, and this is going to be done on blood auger to confirm that the culture was pure, and then we're going to put it into our walkaway, and it's going to give us a readout. Our printout, we did confirm that we're working with Klebsiella pneumoniae, and it flagged for a very rare biotype. So on the top here, we're going to have all of our biochemical tests, and then on the bottom, we're going to have our antibiotics, which we'll get to later. So our biochemical tests, urease positive, VP positive, citrate positive, and indole negative, which are all confirmed and are usual for Klebsiella pneumoniae. Now it gets a little weird when we get into our sugars because Klebsiella is known for loving all sugars, especially the weird ones like inositol, which more pickier eaters like E. coli won't go for. But in our printout, we've got all negatives. And so that is pretty peculiar, except for our glucose. And so I did a little bit of research, and it seems like this is less of a fluke and more of a confirmation for a very rare biotype, just since our biochemical tests do line up with the regular Klebsiella. Let's move on to our antibiotics. Now we've got our ampicillin that is resistant, which is normal for Kleb because it's always going to be resistant. And then we've got resistance to our CFT, our CFC, and our CPE, which is our third and fourth generation cephalosporins. Now let's move to the bottom here because this printout says that we have an ESBL positive, which is our extended spectrum beta-lactamase, which is an enzyme that is produced by Klebsiella pneumoniae that breaks down and disables certain antibiotics, like our third generation cephalosporins and sometimes our fourth. But there is a CP susceptible antibiotic, which is very strange because normally when we have an ESBL positive organism, you're going to see resistance all across the board for your cephalosporins, and this seems to be the exception, which also lines up with our very rare biotype. Next, we're going to move on to our susceptibilities. Again, CP is pretty odd if the rest of our third and fourth cephalosporins are resistant. We've got our CZA, which is our cleftolozane tazobactam, and that's a powerful combo antibiotic that is designed to tackle tough gram-negative infections, especially when there is a resistant problem. We're going to have our carbapenems, which are still effective, and then some other drugs. And those are going to be some broad options that are still available. Um, Again, our isolate might carry a functionally suppressed or variably expressed beta-galactosidase gene, enough for the MacConkey, but not enough to trigger the ONPG positivity under panel conditions. That plus the rare biotype suggests that it could belong to a lesser known genogroup worth deeper characterization. Going to talk about where we normally find Klebsiella pneumoniae, and this is going to be an opportunistic bacteria, so this is not going to infect your healthy individuals, and it's going to be found in hospitalized patients, elderly people, cancer patients, and it's normally going to infect the lungs, the urinary tract, and the blood. And so the lungs, this is going to be found in patients that are using ventilators, and same with the urinary tract, patients who are using devices like catheters. And when we find it in the blood, normally it's secondary to our lung and urinary infections.